Je vous propose de vous installer, on va démarrer. Alors, quelques mots peut-être pour introduire un peu l'événement d'aujourd'hui. Donc c'est un événement, c'est une séance qui va être donnée par Paul Nurse dans quelques instants, prix Nobel de médecine, qui se fait dans un cadre particulier qui est le cadre des Nobel Prize Dialogue. Donc il y a un événement qui se déroule ce soir à Beaux-Arts et qui est un événement qui touche, je pense, à un enjeu extrêmement important. Le titre de l'événement, c'est Fact and Fictions, the Future of Democracy. Et c'est un événement qui essaye de mettre en discussion, de mettre en regard les liens qu'il y a entre science et démocratie. Et je pense que, en tant qu'université, c'est vraiment un thème auquel on est particulièrement sensible et qui s'appuie en partie sur un texte qui a été coécrit par Paul Nurse et son collègue Ben Ferriga, qui est un autre prix Nobel qui donne à ce moment même un exposé analogue à la VUB, qui est notre université sœur, qui ont écrit ce texte qui disait qu'en fait, les liens entre science et démocratie sont importants et s'appuient comme ça sur trois grands piliers. C'est que c'est difficile de développer la science, de développer la pensée critique, la pensée libre dans un contexte qui ne soit pas démocratique, et que donc un fonctionnement démocratique donne un petit peu un terrain fertile dans lequel la science peut s'exprimer, et en retour, la démocratie peut bénéficier de l'ensemble des avancées scientifiques et techniques, et que le développement des fake news, donc cette espèce de vacillement qui peut s'exercer sur la vérité, eh bien, en fait, menace les deux, menace à la fois le développement scientifique, menace à la fois le système démocratique, et menace en réalité des conditions d'un progrès collectif, d'un progrès humain. Voilà, donc ça c'est le cadre un petit peu plus large des discussions de ce soir. Et en marge de cet événement, nous avons été contactés par le Nobel Prize Outreach qui organise cet événement pour organiser à l'ULB et à la VUB ces conférences et exposés qui seront suivis d'un petit entretien plus particulier avec Paul Nurse. Et je voudrais vraiment remercier la Fondation Nobel et Nobel Prize Outreach de nous avoir donné l'opportunité d'organiser cet événement. Je pense que c'est vraiment très riche pour celles et ceux qui sont ici. Et voilà, donc je vous souhaite un très bon cours et je vais passer tout de suite la parole à Adam Smith, qui est le conseiller scientifique de Nobel Prize Outreach qui organise l'événement de ce soir. Voilà, merci à toutes et tous et je vous souhaite une excellente séance.
Merci, Marius. Thank you very much indeed. And as Marius has so beautifully introduced, this is in connection with this evening's event at Beaux-Arts. The event is unfortunately sold out, but you can follow it online. It lasts for 2 hours, from 7 o'clock to 9 o'clock. And if you just go to nobelprize.org, you can watch it there. You can sign up to be sent a reminder to switch on at 5 to 7 this evening. In connection with these dialogues that we hold around the world, we love to hold these lecture events where we bring Nobel laureates to meet mainly the next generation of scientists. And we think a very important part of this is the conversation between you and the Nobel laureate. So Paul is going to give his lecture on what is life, and then there's going to be a long period for you to ask questions. So please, I encourage you to use that and ask lots of questions. I'd like to thank our event partners who make all this possible. You see them listed there: 3M, ABB, Capgemini, était, H2 Green Steel, and Scania. And without their support, we can't organize any of these events. And also, I'd like to thank the European Research Council, who together with us are organizing this evening's event, and the Embassy of Sweden for making things possible. Paul Nurse, as you know, is 2001 Nobel laureate in medicine. He discovered molecular mechanisms that control the cell cycle, and he has had many positions. He's been, for instance, president of the Rockefeller Institute, president of the Royal Society, and now runs and directs the Crick Institute in London. In today's lecture, he's going to talk about what is life, which is a pretty vast topic to address. So, over to you, Paul.
right. Oh, that's better. I don't have to project my voice quite so much. Adam was right. It's a big question. In fact, one of the most fundamental questions in science, certainly in biology. It's very easy to ask, not so easy to answer, but I'm going to have a go at answering it. Or rather, not answering it, but giving a direction of travel for thinking about it. That might be a more honest position. There's an extraordinary diversity of life on our planet. It's all around us. Bacteria, fungi, plants, animals, ourselves, of course. What is it that links all these different life forms together? It's a long time ago, but I think it was a butterfly that first started me thinking seriously about questions like this. It was early spring, about this time of the year. I was about 12 years of age, and I was sitting in my garden in northwest London when this yellow butterfly flew over the fence. It's a brimstone yellow. You see it, including in Belgium, in early spring. It turned, it hovered, it briefly settled, a shadow disturbed it, it took flight, it disappeared over the opposite fence. Not very exciting, maybe, but it made me think, that intricate, perfectly formed brimstone yellow butterfly, both utterly different to me, yet somehow familiar too. Like me, it was obviously alive. But what did it mean to be alive? It could sense, it could respond, it seemed full of purpose. And I found myself wondering, what does it mean to be alive? Which is, of course, the question, what is life? Now, I'm not the first person to ask this question, believe it or not. It's been asked by others for many years. This is the title of the title page of a book. One of them, quite a few books written by different authors with this same title. This one was written in 1944 by the physicist, not biologist, Erwin Schrödinger. His book was influential at the time and in the decades after it, including for me when I first read it, which is now 50 years ago. He considered aspects of life, which I will briefly allude to later. The way I wanted to approach it was to think about 5 of the most important ideas in biology, at least for me. I want to describe where they come from, which is quite illuminating because some of the fundamental ideas we have to think about are not actually very recent. They have their origins deep in the past. So I want to describe where they come from, explain what they are, and then at the end I'll try and pull them together to develop some principles for thinking about life. And the first of these ideas is the concept of the cell. I call it biology's atom. The cell is the basic structural and functional unit of life. They were discovered by a polymath scientist called Robert Hooke, and he published a book in 1665 with this illustration here, the one in black and white that you see. It was discovered in the city I live, actually, at least at the weekends, in Oxford. And he published this picture in a book called Micrographia. And he used a razor to cut slivers of cork, examined them under an earlier microscope, something like the one I put on this slide here, and he saw a neat array of boxes, which he called cells, after the Latin cella, which is the word for small cubicle. And that's shown here in this drawing, and I put a modern scanning electron micrograph of the same material to show you basically the same. It's true the microscopes are a bit different, but basically the same. Now, a few years later, there was a Dutch draper not so far from here in the Netherlands called Anthony van Loonhoek. He was working in Delft, and he scraped between his teeth and put that under a microscope, an even simpler looking microscope than this one, and discovered single celled life. Here's his picture, I think sent about the 1690s to the Royal Society in London. He sent 1,000 letters to the Royal Society, which are all archived there. And this is what he saw under the microscope. I want to draw your attention to what I find the charming figure B to C to D, because this bacteria was obviously swimming under the microscope, and it does a sort of loop the loop during that process. Leeuwenhoek was very disturbed by the discovery of microorganisms between his teeth, because he was very proud of his dental hygiene. And the fact that there's life there, he found rather disturbing. He was a close neighbour in Delft to the painter Vermeer, who will be familiar, I'm sure, to many of you. And Vermeer, unusually for a 17th century, or for that matter, any century painter, painted two pictures of a scientist. This is one of them. One is in the Louvre and one is in Frankfurt, actually. And I like to think this might be Leeuwenhoek. This is, I think, he painted an astronomer and a geographer. I forget which one this is. I like to think it could be Leeuwenhoek. I have to admit to you, there's not the slightest evidence in favour of that, except one thing. When Vermeer died, Leeuwenhoek was the executor of his will. So it is possible that he did know Leeuwenhoek, but there isn't any direct evidence of that. Now, over the next 2 centuries, 1700s, 1800s, it became clear that all living organisms consist either of a single cell or, like us, a collection of cells. And this conclusion was nicely stated by Theodor Schwamm, a German scientist in the first half of the 19th century. And he wrote in German, translated here, "We have seen that all organisms are composed of essentially like parts, namely of cells." Now, as well as cells being the basic structural unit of life, which is what this sentence implies, they're also the basic functional unit of life. What do I mean by that? Well, it was nicely expressed a decade or two later by the founder of pathology, another German scientist called Rudolf Virchow, where in 1858 he said, "Every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life." In other words, the cell is the simplest entity that unambiguously, and I will be distracted by viruses a little later in my talk, but unambiguously we can say is alive. And that's why I tend to refer back to cells, because if we want to understand life, we have to understand what it is about a cell that actually makes it life. He also said, he wasn't the first to say it, but all cells come from cells. Omnis cellula est cellula. And this means that cells arise through the division of pre-existing cells. And in the final sentences of my talk, I'm going to refer back to that in a slightly unusual way. So cells are interesting. But if I fail to convince you of that, let me show you this picture of a sperm and a mammalian egg, and remind all of you in this room, you once looked like this. You were all once a single cell. And if you're no longer interested in my talk, there's 2 doors here. You could leave now. Because this, I think, we were once a cell. I mean, we're of course more complicated now, at least we like to think we are, but we were once a cell. So, as I said, simplest entities that unambiguously exhibit the characteristics of life. This is a modern picture of a cell, artificially coloured. Cells are bounded physical entities separated from their environment. I use that sentence carefully because I want to exclude computer games in any discussion we have about life, because a bounded, separate physical entity, it helps make that distinction. It's surrounded by a semi-permeable lipid membrane which separates cells from their environment but allows good communications with that environment. It's important because this separation is needed to allow order to be built up inside cells at the expense of increasing disorder outside the cells. And in this way, the life does not contravene the second law of thermodynamics, which often worries physicists who think about life, because they worry about it because it seems to contravene that second law. It worried Schrödinger, and I'm just going to put a sentence, the top sentence here, that indicates that. He was astonished by an organism's ability, astonishing gift he calls it, of controlling a stream of order on itself and thus escaping the decay into atomic chaos. A little bit more sophisticated in the sense what he was struck by was the fact that the inheritance from one cell to another maintained that order. And he found that unexpected and extraordinary, and postulated a sort of code script that was inherited from cell to cell. He wasn't the first to talk about code scripts. Actually, as we shall see, it's implicit in the foundation of genetics, and we'll see that in a moment. But another sentence here, which is a good introduction to my next topic, is he refers to chromosomes and says he proposes these contain some kind of code script, the entire pattern of the individual's future development and function is somehow encoded in those chromosomes. A nice transition then into the gene, my second idea. The gene, which explains the mysterious phenomenon of inheritance. This is the idea of the gene as the basis of heredity, excuse me, has its origins with the work of the Augustinian monk Gregor Mendel. Here's a photograph of him. He was abbot of Brno Monastery, an Augustinian monastery, a teaching order in what was then the Austro-Hungarian Empire, is now the Czech Republic. And he published only 2 papers relevant to genetics in 1866, I think it was. And he tackled the problem of inheritance by carrying out crosses of pea plants with different characteristics and analysing the outcomes in the thousands of progeny produced in these crosses. He was trained as a physicist, actually, originally, and so took a sort of quantitative approach to biology. Darwin also did similar crosses, but was much more qualitative in his thinking. I found that when I first visited the monastery, which was in 1981, in the middle of the Cold War, this is what his garden looked like, where he did his pea. It has a sort of dark look about it. It was in the middle of winter, I have to say, then Czechoslovakia. But what I want to draw your attention to, it was actually quite a large research garden. This wasn't small. And next to it, which you can't see because it had been demolished, was a greenhouse of similar size where he was doing his research. The reality was this monastery was a research institute. This was just a surprise to me. You don't think of monasteries as being research institutes. But in fact, the monks there were studying all sorts of things. He was doing genetics. A map of the moon was made there. Meteorological experiments done, associating storms with reduced pressure. And Mendel was involved with a lot of these things. It's something, we're so embedded in what we think how research is done. It's just to remind us it wasn't always quite like that. It was related, of course, to the fact they were a teaching order and so quite academic. I should say, though, Mendel wasn't good at exams and never passed his exams in physics in the University of Vienna. Never passed. So if any of you out there are having difficulty with your exams, don't despair, it isn't the end. Now, what Mendel did was to cross, as I've said, these peas of varied characteristics, plants of different heights, for example, as you see here, different flower colours, different seed shapes. And he famously observed simple ratios in these characteristics in the progeny produced by these crosses. I'm not going to explain these to you because I don't have the time, but you will have come across them at school or at university. Ratios like 3 to 1 and 9 to 3 to 3 to 1. Just nod at me and then I'll feel I can move on. And this led eventually to the idea of Mendelism. I have to say Mendel himself didn't quite get there. We call it Mendelism, but he didn't quite get there. It was really better explained 30 or 40 years later, when his work was rediscovered by three plant breeders, including ones working in the Netherlands, by the way. And Mendelism, the idea is that heredity is based on pairs of indivisible particles or factors, one of which is inherited from one germ cell, the other from the other germ cell, pollen or an ovule in the case of plants, sperm or eggs in the case of animals. These factors were subsequently found to be linked to the chromosomes, the one that Schrödinger mentioned in his talk, in his book. And I find this quite interesting because Mendel's work was ignored completely for 30, 40 years. But in between 1865 and about 1902, chromosomes were discovered. And this is a late 19th century onion root tip showing chromosomes. You know, these are these black strings you can see there separating. Actually, these, I first saw cells and chromosomes doing an onion root tip squash exactly like this when I was at school. But the point about the chromosomes is that scientists began to think the way they behaved suggested that they could be responsible for heredity, because you had strings that looked similar that were being separated every time a cell divided. So then it was natural to take Mendel's more abstract analyses, just counting numbers and saying there's particulate inheritance, to an actual physical manifestation of it, looking at chromosomes under the microscope. And I think it was that linkage of the abstract with the concrete that allowed this work to get a much stronger currency. Anyway, this was very important, particularly in the 20th century, for genetics, because these Mendelian hereditary factors are what we now call, of course, genes. In the 1940s, Oswald Avery, working in the Rockefeller Institute in New York, where I was president when it became a university, in 1944, they showed that genes were made of DNA, or deoxyribonucleic acid. There were 3 researchers doing that, and one of them was called Matt McCarty. And when I became president, which is now about 20 years ago, I had to have an introductory cocktail party, meeting all the faculty. And this little old man came up to me and shook my hand to welcome me. And I asked him his name, and he said, "My name is Matt McCarty." And I said, "You aren't the Matt McCarty?" And he said, "Yes, I am the Matt McCarty." That was my introduction to Rockefeller University. Now, that isn't so well known as those that discovered the structure of DNA. And the structure of DNA as a double helix was established 10 years later in London by Rosalind Franklin, Ryan Gosling and Maurice Wilkins, and in Cambridge by Francis Crick and Jim Watson, who did the modelling of the experimental work from London. And this is the chromosomes from the nucleus of a cell being unraveled to identify the double helix. And Crick and Watson famously argued, having developed this structure, that a double helical nature of it could explain heredity through the linear sequence of bases making up the DNA and the pairing between complementing bases that requires. Adenine A pairing with thymine T, cytosine C pairing with guanine G. Here's the double helix again, and I want you to imagine the double helix as a twisted ladder. It's just easier to imagine it untwisted, just a ladder with rungs made up of A to T and G to C. And when these links are split, you can produce two templates which will then generate an exactly complementary copy, and that will precisely replicate the DNA code script every time a cell undergoes reproduction. And as you heard in the introduction, that's actually what I've spent my life trying to understand, how the cell cycle is controlled, not only the control of DNA replication, but of mitosis and cell division. Importantly, it explains the perpetuation of Schrödinger's code script and the permanence of genes between generations, because they're constantly copied. And the information of the code script is precisely copied every time a cell divides. Crick went on to propose what he called the central dogma. It's a typical Crick term, by the way. I met him quite a few times earlier in my life, and he had this ability to come up with great ideas and give them great names. This is the central dogma, you know, and explains how the DNA encodes the structure of proteins that I will talk about in a moment. And this describes the sequence of bases making up the gene in the DNA. It's copied to ribonucleic acid or RNA. That acts as a messenger between the nucleus and the cytoplasm. DNA is in the nucleus, protein synthesis based on this in the cytoplasm. And each gene essentially encodes one protein. These discoveries laid the foundations for what we know as molecular biology, and they are critical for understanding life. Central to this is this genetic code. Heredity is written in a four-letter code, 4 bases, A, T, G, C. And the information is stored in a linear form. Now, this way of storing information is the same as the sentences that I'm speaking to you today, or the sentences you read in a book, or bytes in a computer. And what it essentially means is that the linear digital storage of information, which is what it is, was invented by life over 3 billion years before the computer age. The computer age is based on exactly the same principle. Now, the nature and function of the gene introduces the next 2 ideas I'm going to talk about, and that is life as chemistry and life as information. First, life as chemistry. That is, how chemical activity and physical forces can bring about life. Now, the idea that the functioning of living organisms is based on chemistry and physics can be traced back to Lavoisier, the father of chemistry, famous French chemist. He was studying fermentation, as so many French scientists seem to do, something to do with wine, of course. And he was trying to relate it to the energy and so on that was present, that was being consumed. And unfortunately, he had his researches terminated by the French Revolution because he lost his head to the guillotine. He was a tax collector. Actually, he was in favor of the revolution. He was a tax collector. And he lost it in about 1794, something, 1794, something like that. I don't know if you know the painter David, who painted a picture of Lavoisier. It's in the Metropolitan Museum in York. It's, to me, the best picture of a scientist of all time. But David painted this, and 10 years later was on the jury that condemned him to death. Very poignant, I think. Anyway, he lost his head, so literally, so didn't take his work any further. But Louis Pasteur, here's a romantic, less impressive picture, I have to say, of Louis Pasteur, who carried out experiments on sugar beet fermentation, which really strengthened the idea that, of the concept that life is based on chemistry. What Pasteur was doing was analysing the fermentation of sugar beet to make alcohol. This was an industry in northern France, and he was asked to do this at the request of industrialists. In other words, he was doing applied research commercially, because there isn't such a distinction. There are distinctions, but many of the practices are the same between discovery, research, and application. And what was troubling the industrialists is because the fermentations sometimes failed, didn't make alcohol, but made acid instead. And acid was valueless, okay, with, it was acetic acid mainly, compared with alcohol. And he just looked at what was going on under his microscope and demonstrated that successful production of alcohol required the activity of a single-celled living organism, yeast. He proposed that the yeast cell produced substances that made alcohol from the sugar, which is derived from the sugar beet, and if yeast was not there, then bacteria would take over the fermentation, and those bacteria would ferment acid instead. So he concluded that as long as they added yeast to the fermentation, everything would be right. I don't know if he got paid for that or not. There's no record that I could find as to whether he did. But what is important for us scientists today is that from that applied piece of research, he concluded something more generally. That is that fermentation should be considered, and this is a quote from him, a physiological act yielding products for the cell. In other words, this is him, chemical reactions are an expression of the life of a cell. In short, we understand life in terms of chemistry. But it's a very, very special and elaborate sort of chemistry. It's based on carbon polymers, as are DNA and RNA, and now we need to consider the carbon polymers that make up proteins, because the substances from the yeast that Pasteur postulated made the alcohol are proteins acting as enzymes that catalyze chemical reactions. And I'm going to spend a few minutes about that and perhaps explain it in a way that makes it a bit more wonderful than reading it in a textbook. Proteins are polymers of amino acids with a backbone and side chains. And the side chains, the backbone is similar, but the side chains, and there's 3 or 4 different amino acids here, are different. So what we have up here, they can have different chemistries and different sizes. Lysine, leucine, tyrosine, aspartic acid, basic, acidic, hydrophobic, bulky, not so bulky. So lots of chemical diversity. And there's 20 of these which are used in life, only 20. And the result of this is a wide variety of chemistry. Here, by the way, it's just to show what the backbone looks like. And where you see yellow R's, that's the side chains that I've illustrated in colour here. Well, I didn't. I think I stole this from Scientific American or somewhere. But anyway, you get the idea. Now, this means that proteins are chains of amino acids. And they have different chemistries, which mean they can fold up in different sorts of ways, depending upon the amino acids being used and the position of the amino acids in the chain. And that gives you different sorts of structures. And here I've turned it into a billiard ball structure. Again, it wasn't me, it's probably Scientific American. And the different colours mean different chemical characteristics. And it's the fact that the proteins express a wide variety of different chemistry, which is very significant, and I want to emphasize here, because DNA does not. DNA is actually dull. It encodes information, but it doesn't carry out a wide variety of chemistry. But the beauty of this is you turn DNA into proteins that can exhibit a wide variety of chemical reactions. These enzymes and other proteins act as chemical machines, and these machines can produce large quantities of different chemicals required for life to function. They can make new molecules, break down molecules in existence, they can recycle them, generate molecular assemblies, and synthesize the polymers of DNA and RNA. They can do physical work. They even make motors and run around on sort of trackways in the cell. They can act in hybrid ways of chemistry and physics, capturing energy from sunlight and to produce energy-rich chemicals that can drive other reactions. It's amazing. I don't think we think about it. It is absolutely amazing. And all going on in a little 10 micrometer sort of a cube. And all these reactions, very different as they are carried out simultaneously close to each other in living cells. This is an example of something I had to learn when I was an undergraduate. Each of these dots is a molecular mechanism. Can you see the red circle in the middle? That's the Krebs cycle, just to give you a reference. There's a couple of hundred reactions on here. There's actually thousands going on in this tiny space. Now, this can only be achieved by compartmentation, so that there's some separation of these different chemistries, one from another. It's not just a fluid mixed up, it's compartmentalized. Now, this compartmentation is brought about by the surfaces and folds of the enzyme, the protein, which can give different conditions, by complexes of different proteins together, which can do subsequent reactions, one after another, or by membrane-bounded cells, going back to membrane-bounded organelles in cells, going back to this picture. Now, I emphasize that because this is always sort of looked at as if it's static. But it isn't static. This is a gigantically dynamic object where all these reactions are going on together and separated one from another. And it's only because we look at them as static objects that we get distracted into thinking that isn't going on. Now, there's a consequence of this. And that is that although this compartmentation can mean that many different chemical reactions can go on close to each other, they've got to act together as a whole. It's got to act together as a whole system. And that can only be brought about if these different compartments have ways of communicating one with another, so they're separate but in communication. And that implies that there is management of information, because communication from one place to another implies information, different from the DNA information that we think about. This is to do with a complex system that is based on information. And that's going to bring me to my next idea, life as information. This is, I thought this would be a modern idea, because everything I've talked about had its origins in the 17th to 19th centuries. Except for the molecular biology. I'm going to talk about complex systems, control and purpose. But I tried to find out where life was first discussed in terms of information. And probably to your surprise, the earliest account I could find, was a piece written by Kant, the philosopher, Immanuel Kant, around 1800. And he wrote a book on moral philosophy, and for reasons that I simply don't understand, there's a 30-page discussion of the complexity of life, which has to be based on management of information, I'm using modern words, and systems. So this is an idea that's been around for over 200 years. Wasn't fully explored, but has been around. Now, this is really important for understanding life, and I'm going to give two examples. One you've already heard about from me, but I've already explained it, so it's easy to refer back to it. The structure of DNA is beautiful, double helix. Although I'm sick to death of biological institutes which have a piece of art based on the double helix. And when I set up the cricket, I said that we are not having a piece of art based on the double helix in the building. And I noticed a few weeks ago, I found one. It'd been smuggled in. I'm trying to get rid of it, but I've failed so far. Now, the structure of DNA is iconic and beautiful, but its real beauty lies in the fact, not in its chemical structure, but that it only makes biological sense when it's recognized, as I've already explained, as a digital information storage device. That turns the chemistry into life, because we have to understand information and its coding. One written in this linear script of 4 letters. Now, the linear polymer, as I've explained, is ideal for storing information. And actually, the encoding bases are on the inside of the double helix. They are, in actual fact, partly protected from chemical change. But just to stress this, the beauty of this, as Crick really argued, is that this very stable information, which is chemically mostly inert, can be turned through transcription and translation into chemical activity through the action of proteins, which are highly chemically active and therefore less stable. So you can have both storage of information and also output of that into chemistry and physics to do the job of life. And so this is turning, if you like, a one-dimensional structure into 3 dimensions, and it turns from information into action. The second one I want to talk about is this machine here. It's about regulation, how you make complex systems work as a whole, apparently with purpose. Now, I don't know if you're like me. I can understand most man-made machines before 1900, and increasingly less as the 20th century goes forward. So I've gone back. This is from a steam engine built in 1896. I discovered it on a boat in New Zealand, I think. And you can understand it. It's a governor, which was invented by James Watt, who was actually inspired by Huygens, who used this to this type of mechanism to control windmills, believe it or not. Anyway, so this spindle rotates. The balls go out. They lift up a valve so you can open and close it. So as the machine goes faster, it switches off steam going into the machine and it slows down. Then the balls go back in again, lets in more steam, and up it goes again. This is a homeostatic mechanism brought about by a negative feedback loop, a regulatory loop. And here's a couple of 2 simple control modules. Negative feedback loop, A to B to C. The build-up of C inhibits A to B, so C then goes down. A positive feedback loop, complete opposite. As C builds up, it promotes A to B. So once you've started doing that, it's an irreversible switch. This type of thinking was applied to living cells to bacteria by 2 brilliant French geneticists, Francois Jacob and Jacques Monod. And they did it, the most brilliant experiments, just simply doing genetics, counting bacterial colonies, simple enzyme assays, and they produced this model, which is the basis of understanding how information management can bring about, as they describe, homeostasis. And these feedback and control modules operate in cells. They're homeostatic. They have generate switches and timers and toggles and oscillators. They translate chemistry models into informational modules. I mean, that's what's going on here. And this can be linked to even generate even more elaborate routines of informational management and processing. And I think that we've barely got to grips with that. I mean, these days you talk about systems biology. This is the theoretical side of it. It's not just collecting observations, which is where sometimes people get distracted. It's trying to work out how it all works together is what is important in systems biology. A metaphor for this is this electronic circuit of linked modules. I know where I stole this one from. It's from EMBO Journal, in this particular case. But an even better metaphor is wetware rather than hardware. This is hardware. Because in cells, you connect these different systems through solution chemistry. So that means you have the ability to change the hardware because it's wetware, and you can connect different things. And this is something else that I don't think we barely got to grips with the versatility of this. So the management of life's chemistry by information allows the behavior of this complex system to act as a whole with overall purpose, to grow, to maintain itself, to divide. And we need that if we are going to reproduce a cell, make a butterfly, maintain our cells. Now, for centuries, the generation of such purposeful behaviors, I'm deliberately using the word purposeful because some people are uncomfortable with it. Happy to talk about that if you want. It was thought to require a designer, a creator, often a divine creator. And it took evolution by natural selection to change this, and this is my last idea. So if I've been talking too long, take a deep breath. It's coming toward an end. This is the final idea. Then I have to put it together. Now, this is evolution by natural selection. How living things come about, how they change. It's a beautiful idea, probably the most beautiful idea in biology. It's mostly, but not entirely, due to Charles Darwin. Has two parts. Life evolves over time, changes, which was not Darwin's idea. That was well established as a proposal before Darwin. And two, the major mechanism for evolution is natural selection, which also wasn't entirely Darwin. It had been in parallel suggested by Wallace, but Darwin had thought about it much more than Wallace. Just dealing with the first, I want to introduce another character to you, which is Darwin's grandfather. The guy over the left there is Erasmus Darwin, and he worked in the late 18th century, beginning of the 19th century, 40, 50 years before Charles. And he and other scientists like the Frenchman Jean-Baptiste Lamarck argued that life evolved. That was Lamarck had a mechanism which was not natural selection, but they both argued that life evolves. But I mention Erasmus because he was such a colourful character. He was English, he lived in the Midlands, he was nowhere near a university, nowhere near a university. Oxford and Cambridge had nothing to do with him, okay? Nothing to do with him. They were a bit degenerate at the time, it has to be said. He was a scientist and a doctor. He earned his money as a doctor, but he also was a poet. And he wrote all his research up in terms of verse. He didn't write papers, he wrote up verse. I got fascinated with him, so I bought a number of his original books. And it's just, 200 pages of blank verse describing the loves of plants and what it means for the different types of plants. He was a Republican, so he refused to be George III's personal physician, which of course was the highest distinction. He wouldn't do it because he was a Republican. He was a proponent of public education for women, which was simply nowhere at that time. You could get private education, but not public education. And indeed, two of his daughters were school teachers in such a school, and he wrote the book about what girls should be taught. And he was a great conversationalist, despite having a terrible stutter. He lived in the Midlands and was one of the founders of the Lunar Society that would meet once a month under the full moon. They were called the Lunatics for that reason. And they included people like James Watt and so on, and talked about science and engineering and politics. Now, that was just a distraction. The evolution of life implies there's a tree of life. And the tree of life implies that living organisms are all related. That's a key point I'm going to emphasize. If life only arose and survived once, that seems likely given the highly conserved chemistry between you and bacteria, for example, then all life on Earth is related to each other. Key thing to remember. This connection was captured in the only diagram in Darwin's Origin of Species, published in 1859. And this is the first tree of life. And you'll see at the top there are the life forms that have survived, and you'll see all the ones that died on the way up. Notice the letters at the bottom. Darwin was not sure whether life arose once or many times. I've just made an argument because of the chemistry that maybe it arose more than once, but what survived is probably only one. And what we see today is based on that single chemistry. He gathered evidence in favour of this of natural selection through his trip round the world in a boat called the Beagle. And I'll briefly explain what natural selection is. It's the consequence of populations of species exhibiting variations caused by inheritable genetic changes. These can be caused by mistakes when DNA is copied. I'm, of course, giving a modern interpretation of it. He didn't know about DNA. Or if DNA is damaged. If the variants influence characteristics that make these organisms more successful, better adapted, they will reproduce more. And if they reproduce more, then eventually that genetic variation spreads through the population. And over time, and this is the argument, when genetic variations accumulate, they lead to sufficient changes in that population such that individuals in the new population would no longer be able to breed with the original population. And that is the origin of a species. I mean, it's a simple and beautiful idea, still useful, particularly in computer programming, for example, where the principle can be used. Darwin came partly to that due to his study of finches on the Galapagos Islands. Galapagos, the volcanic islands, and of recent origins, and they had a whole range of finches that are only found there, and they were specialized for different purposes. The one at the top breaks nuts. The one at the bottom finds insects under bark, and you can see how they are better adapted. You'll see on the right another sort of specialization with pliers which do different jobs. You see there's pliers here that at the bottom which are used for electrical things, at the top which are used to unscrew nuts. But the difference between these two are that these were produced by natural selection, and these were created. Okay? These were intelligently designed, and that is, of course, the distinction between speciation by artificial selection and design. So the finch's beaks could be produced without a divine creator or designer. You need three things for this to work. Reproduction, obviously, a heredity system, obviously, and a heredity system that can exhibit variation. And with those three things in place, you can get diversity of life. If that wasn't clear, a very simple example. Imagine a yellow cell. The yellow coat colour is encoded by a gene. Imagine a mutation, so you now make a red coat. Imagine the red coat is not attractive to a predator. Then what will happen is the red-coated cells will outcompete the yellow ones. And so you get natural selection. And notice how it's based on ideas that we've already discussed, the idea of the cell and the idea of the gene, as well as the idea of the system. So it all comes together. This is a very important idea. So much so that the 20th century geneticist Hermann Müller used it to define what life is. He said, living things have properties which allow them to undergo natural selection, that is, to evolve. So this is central to this, and I'm going to consider this in a minute or two. But first I want to say goodbye to Charles Darwin with this brilliant sentence, which is the last sentence in The Origin of Species. Do you mind if I just read it out? Whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Why can't we write scientific papers like that anymore? You can read this 200 years later, and it's amazing. I can barely read what I write myself 2 years later. And by the time it's been gone through reviewers and edited and so on, all interest is lost. We've lost something. We really have lost something. He's clever. Notice he refers to gravity. Why did he refer to gravity? Because the greatest of scientists of all time at that time was of course Newton. And Newton was the paragon, and he was saying, not without actually saying it because he was subtle, that biology can have laws as important as physics. Because then physics, the queen of sciences, and as Isaac Newton, and this should be seen in the same way. Now finally, before I sum up, let's have a brief comment about viruses. Viruses. From which I've been suffering, actually, so I'm not very sympathetic to viruses at this moment. Now, all right, I'll tell you what I'm going to say about viruses. Viruses have a genome, they have DNA and RNA, they undergo evolution by natural selection, as we've seen with how many variants COVID-19 produced. They thus pass Muller's test. But they can only reproduce when they're in the cells of other organisms. So they are completely dependent on another organism. They hijack the machinery of that organism to copy itself and to make more viruses. So in thinking about this, a virus is alive when it's in a cell. Maybe it's an inert chemical when it's outside. That's maybe one way to look about it. But I'm going to take a slightly different approach to this. Because in thinking about this, I think we need to realize that other forms of life are also to a greater or lesser dependent on other living beings. And that's something we rather ignore in thinking about viruses. The many parasites that live on or inside the cells or bodies of animals, plants and fungi are clear examples of this, of course. Their dependencies are less than a virus, but they're still dependent upon that other organisms. Even we are. There are certain amino acids we cannot make, and microbes in our body make them. And of course we eat, essentially, food that comes from other animals. So that means there's a graded spectrum of living organisms where different life forms have varied dependency on other life forms. In the case of virus, that's very strong, that dependency. Whilst in other living organisms, cyanobacteria, for example, which capture energy from the sun, that dependency is weak. But I'm going to argue that different life forms, these different life forms are all should be considered living because they evolve by natural selection. But they also are highly interacting with other living things. And that means life on Earth is fundamentally connected, both through being related as a result of evolution by natural selection, and also through these deep relationships and interdependencies. Again, something that isn't often stressed, I would say, in the textbooks. Okay, so I'm going to finish now, and I'm going to quickly go through where I think we've got to in establishing principles to understand life. I need to warn you, it does not lead to a neat dictionary-like definition of life. Number one, living things are independent, bounded physical entities. The basic unit of life on Earth is the cell. They're separated from their environment, in communication with it. They can generate order and organization. Organisms like us are multicellular aggregates of cells. True. This is Muller. Living things possess the ability to evolve by natural selection. To evolve, you have to grow and reproduce, and you have to have a hereditary system which exhibits variability. It's a principle. 3, living things are chemical, physical, and informational machines. They construct their own metabolism, and that metabolism is used to maintain themselves, to grow, to reproduce. It would be a long entry in the Oxford English Dictionary to put all of that together, but these are the principles. We can add a few more, which is related to life on Earth. Life on Earth is based on carbon polymer chemistry. They produce nucleic acid, which are information storage devices, and proteins, which are chemical and physical machines. Life is based on information management and information storage in a linear form encoded in the polymers. And life on Earth is related by common descent and closely interconnected and dependent upon other living things. Now, should we ever encounter life elsewhere in the universe, which probabilistically it probably exists, I suspect it will be based on similar principles. The chemical details could be different, I can imagine that. But I can't easily imagine life without polymer chemistry of some sort, because of the ability to store information and turn that information into active chemistry. And I think that's one thing that I'd like to communicate. It's often not stressed enough. But let's return to our own planet briefly to the end, because this is the only corner of the universe where we know for certain life does exist. We know that. The life that we are part of here on Earth is it's amazing, bewildering diversity, and we are making sense of it. And I want to say our improving understanding has potential for our culture and civilization, and also to improve the lot of humankind. And it shows that all living organisms we know of are related and closely interact. We are bound by a deep connectedness to all other life on the planet, to the bacterium, the fermenting yeast, the pea plants, and our yellow brimstone butterfly. They've all accompanied me on this talk. They're all part of the biosphere. They are all life's great survivors, if you like, the latest descendants of a single immeasurably vast lineage that stretches back through, and I mentioned this at the beginning of my talk, through an unbroken chain of cell divisions into the far reaches of deep time. We are connected through these cell divisions, through our germ cells, and it goes back through generation to generation to generation to the beginning of life. I mean, it's extraordinary. Until I wrote this book, I'm going to tell you about it in a moment, I'd never thought of it like that. But we're not only connected, we're connected back through 3.5 billion years. However, as far as we know on Earth, we're the only life forms that can see this connectivity and reflect on what it means. And because of that, I argue this gives us human beings a special responsibility for the rest of the life on this planet, made up as it is by our relatives, remember. Some close relatives, the great apes, some hugely distant, but all our relatives. We need to care about it, we need to care for it, and to do that, we need to understand it, which is what I've tried to do in this talk, and if you enjoyed it, I've written a book too, and it's in French as well. Thank you very much for listening.
Thank you very much indeed, Paul. I have a question for you, which is that you founded and direct the Crick Institute. Oh, yes.
Yeah.
That keeps you busy. You're on numerous academic and other boards. You travel all the time. You're an advisor to governments. How did you find time to write a book?
Well, the advantage of writing things is you give jobs to other people. I'm actually perfectly serious. I mean, the problem with leadership is when they think people don't make decisions. Leadership and running things basically is assembling people who are as good as you or not better.
Yeah,
letting them get on with it.
You're remarkably good at finding good people, actually. What's your secret, do you think, for that?
My nose. I pay a lot of attention to recruitment people and their personality and character to see whether they can interact well. Yeah. And I've always made time for my own research and for thinking about things like this. So half my life is concerned with doing the stuff you just said, but the other half is running my lab and thinking. If you run a university, you can't do that, because it's just too difficult, really. It's really difficult. It's overwhelming, which is why often jobs rotate round in time. But in my case, I can give jobs to other people.
Okay, we have microphones.
I'm a bit deaf. You'll Adam will interpret.
Put your hand up. Yes, please.
Yeah. Thank you so much. Hi. Thank you so much for the talk. It's really inspiring. I have actually two questions. The first one is more practical, I guess. So I'll start with it. So in this concept, would heterotransposons fit as a life form?
I think so. I think for me, the key concept is the ability to be able to evolve by natural selection. And if you centre on that, then it becomes quite inclusive. It means I have to include viruses. And I'm reasonably comfortable with it because, the human mind loves taxonomies. It likes to split things up into sort of indivisible bits, whereas the reality is a much more blended reality out there. And sometimes we get too excited about distinctions. And so the standard, school essay for a 17-year-old is a virus alive. Is trying to trick them into being on one side of the fence or another. Forget about the fences. Second question.
The second, it's maybe something more philosophical, but it's something I've been wondering about. Bringing a little bit the topic of democracy. When I watch videos of people talking about why they defend one side or the other, it's like in everybody's mind, we have kind of a different universe. They see life in a different ways. Even from myself, how I see life, it's like multiple universes, and each person has, And then I kept wondering that we may be biased and limited by our, brain or our constitution, our physical constitution, which would mean also that the way we see life and we understand what is life is also limited by our body and our brains. And if we would imagine that there is another conscious being, another planet in the universe from another constitution and, other kinds of brains, and maybe what life is for this being would be different than what life is for us. So I would just like to hear what you have, if you have some thoughts about it.
Well, there's about a month's worth of questions.
Sorry.
Firstly, I confined myself to thinking about the thinking sensitive, which is difficult enough. So I don't quite get into the mind and thinking. Sometimes thought about doing it, but I put up the white flag and that's it. Might touch on it. But that isn't the only thing you said, so I can't comment so much on, well, I could comment, but not very usefully. But I think one thing that's worth, I could comment on is your notion that, which is very true, that thinking about these problems, we are limited by our ability to think. And I think in this question about what is life, we've been limited often in the sort of technical explanation of it. They just describe what life looks like. We breathe, you know, this sort of stuff. You know, when I was at school, there were 9 characteristics, there's probably 7 now, or 11, I don't know what. But, and I did try to think of what the commonalities and principles were, and principles that you could apply elsewhere in with a completely different structure of chemistries, and it still might apply. But the point you make is sure, we can only think with the brain we have. And it may be inadequate, but there's not much more we can do about it. We might end up talking about artificial intelligence sometime. You know. Will that help? Might it? Might it not? I don't know. So our view of the world is dependent on our ability to think and perceive. And that's all we can work with.
Thank you. Lovely question. Thank you very much. Yes, you are next, please, and then you.
It's open?
Yes, it's on. It's on. It's on.
Thank you. Thank you very much for your lecture. Okay, you explain clearly what is life. But my question, because you start from the cell, I mean, it's already very, very complex, the cell, and it's very organized. You explain that, we understand that, the information. But what before? I mean, what the origin of life? I mean, because, okay, if you put the amino acids all together, and you put some pressure, some temperature, I don't know, will you make a cell? What is the beginning, I mean, for the cell?
Well, it's a very important question. And you'll notice I didn't mention it at all. And the reason I didn't mention it is I don't know. I really don't know. I think it's quite mysterious. The solar system was formed about 4.6 billion years ago. The Earth formed and cooled down and was maybe cool enough to imagine life forms, an environment that wasn't completely alien, maybe 4.1 billion or something like that. Mm-hmm. The earliest fossils are between 3.5 and 3.7 billion. Yeah. So within 500 million years, we had a life form, even though it took another 2.5 billion years to actually make multicellular organisms, which for me is trivial compared with making the first cell. And I think that's something we have to just ponder on. I mean, it is extraordinary. One thing I would say, the fossils that we see from 3.5 billion years ago, we shouldn't assume that they are fantastically efficient. When they grow and reproduce, they might mess up a lot of the time. So evolution might be extremely slow, but perhaps the cells wouldn't, many of them wouldn't survive. And if you're a multicellular organism, you've got to get to a state where all cells are mostly going to survive, because otherwise you can't support it. So that could be a reason. But 2.5 billion years, it's or even 3 billion years. So how do we get life? Well, we have in Brussels, I never say his name right, the hypercycle. Yeah, Piaget. People have thought about and come up with ideas that help. But it's very difficult imagining what was going on 4 billion years ago on the planet. And none of it is entirely satisfactory for me. Some people say to get out of it, oh, it came on a comet. It happened somewhere else. That's just a cop-out, you know. But, you know, they say that. There'd been a divine creator. No, can't be excluded. And once the clock was set running, then the whole thing just evolved. Because we don't need one now, I would say. But, I mean, perhaps you know, you could make a case. I sort of think that there was, and there's all sorts of theories and ideas out there, I sort of think in the volcanic vents and so on, that little rock cavities might have actually formed a primitive cell that didn't have to be made by the cell. And so you didn't have to create something there, you could just provide a spatially ordered structure which could spread. And then you'd have selection, if you like, for things that could spread. And that isn't a good answer to your very good question, but I think it could be some combination initially of not structures, the structures come from somewhere else, but just developing the chemistry. But bluntly, I put up the white flag, so I don't consider it. The publisher of the book wants me to consider it in a second edition. And I'm trying to find a way to wriggle out of it. Thank you.
Please.
Thank you for your talk, Paul. I'm over here. I don't know if you recognize me. I used to work for you. It's been a year now. Well, I'm the guy that always used to. No, I always used to ask questions. So I've got two questions. The second one relates. Yeah, the second one relates to what you were just discussing now, but I think this one is more interesting. Okay. Do you think that maybe we're not being ambitious enough? You know, the physicists have been chasing this theory of everything. And why aren't we going for a definition of life that can fit in the dictionary, as you say? And just to add a bit more to that question, I mean, how far are we? Because we have the central dogma that tells us basically that information is contained in DNA. When the Human Genome Project was being promoted and being carried out, the idea was once we have the sequence, we could more or less reconstruct from the sequence the organism. It's been a few decades now. We haven't quite achieved that. But is that so? My point is that the paradigm that will bring us a theory of everything in biology, or should we look somewhere else?
Let me consider the second thing.
Yeah.
Many people didn't say it would provide everything. It's often stated what you just said, but geneticists wouldn't say it because they're used to thinking that you have to produce an organism and that has to do all the things we have here. Critics of it say, oh, they said they would solve everything. I don't think they ever did. What they actually said, I wasn't involved. Well, my lab did sequence the third eukaryote, it's true, because it was so small we could do it. But what was actually said is you need to have this information before you can progress. So I think it's been caricatured, it's my honest view. I'm not saying some people might not have said that, but it was not the dominant view. But without that information, we couldn't make progress. With it, we then need to understand how all of that's turned into the amazing things I was talking about. The systems and the regulation, and we're hardly touching the complexity of it. So I think that, to agree with you, this is a big project. We needed the sequence, needed to have central dogma, because that's part of it. Now we have to work out how it all works together. We always knew that. And it's really challenging. So when people say, oh, aren't all the problems solved? Of course they're not, as you were arguing. It's a fantastic time for discovery. It isn't easy. Life isn't easy to understand. Now, the theory of everything. I read these books on the theory of everything, and I come out sort of knowing nothing in the end, it seems to me. And my daughter is a high-energy physicist, particle physicist, and so it's sort of immersed in this stuff. And she sort of agrees with me, really, on a much firmer basis. I'm a bit more modest, I suppose, in thinking, you know, I like to make smaller steps. And I do find that, particularly in the life sciences, when you have grand theory, you know, what is it to think? You know, how does a brain work? And we think we're going to have, you know, 10 years of work and we know how brains work. It's all hooey. The biology is bloody difficult. It's really, really difficult. And we have to have humility in approaching it. So, actually, I'm interested in big ideas, some big ideas in what I said today. But let's be humble about it too. So I'm half agreeing with you, half disagreeing.
Okay, thank you for that. And then this more comment, and interesting to hear what you think. So there's this idea that life might basically just be Maxwell's demons all the way down. So what I mean by that is, if people aren't familiar with Maxwell's demon, it's this thought experiment where you have an entity that can basically tell the energy of particles and can sort them between two compartments. And essentially, if you look at what life is doing, so take for example the cell. It's a compartment. The membrane sorts what goes in and out of the cell. We can do this on a molecular level, even with the enzymes. I don't know what you think about that. Maybe at the beginning of life, all you needed was a demon that could select the molecules that would then go on to evolve. And actually, I think you can extend the analogy to evolution, because you're selecting mutations that benefit the organism versus mutations that don't. So you have a sorting process again there as well.
I'll tell you what I think about it. I think that understanding things like life, you have to understand it at the appropriate level of organisation. And when people argue, let's return all to Maxwell's demon, they are not asking the question at the appropriate level. And that if you're a particle physicist, you will ask questions in this sort of bandwidth, and it's appropriate. When you come to the cell, it's a completely different level of complexity. When you come to our minds, we're in another territory altogether. So I think in terms of making sense of the world, we usually, if this is a problem with, we're looking at the sort of bandwidth here, and we are most likely to be successful if we stay within that bandwidth. And trying to reduce it, to the laws of particle physics, I just don't think helps us at all, actually. I know some people argue it, and this gets us into the questions of holism and reductionism, and I think this is also an unuseful distinction to make, because obviously if you're trying to understand a complex system, you've got to look at it as a whole. And obviously the components that make it up, the reductions, are attributed to that. And for some reason we like to make a big fight over it. It's just, it just depends on people's sort of approach to the problem, where the emphasis is. So I think Maxwell's demon is a demon. And I leave it alone.
Thank you.
Okay, thank you.
Please.
Hello, thank you for the amazing lecture, first of all. My question is, because we talk about how all life is related to each other, and my question... Okay. I have a question on relatedness of the things that we have that are not alive, such as rocks and minerals, because we're also talking about how billions of years ago there was the first life, and before that there was nothing. And so let's consider that there is no outside intervention in any case. So we're talking about just rocks and planetary things, and those non-living things turning to living things. And I find myself thinking about those for example, some of the people that I talk, they consider planets as non-living things, or we observe the planets and say that there's no life on this planet, but we don't talk about the potentiality of something that's not alive coming alive. What is your opinion about that?
Paul, I'm sorry, we're actually out of time, so this needs to be your last answer, I'm afraid, at this point, probably.
Well, some people think, you know, Lovelock, the Gaia hypothesis, that you should consider, you know, the whole planet in some sense alive. If you're interested in this, there's a philosopher called Alfred North Whitehead, who wrote exactly about this. He's almost incomprehensible, but I mean, in the 1930s, 1920s, 1930s, who was also of the opinion that in some sense rocks could be considered alive. Now, I don't happen to agree with him, but you might find it interesting to read about. I thought of having another look at it in my old age, because I read it last when I'm about your age, and see what I think today. But I'm more traditional, you know. I think living things are alive and rocks are dead. Maybe I'm wrong.
And what was the name again?
Alfred North Whitehead, Whitehead, a very academic philosopher. There's one more quick question.
Sure.
I just had a simple question. What's your opinion on speciesism?
On speciesism?
Yes.
The question is, what do I think about speciesism, by which I assume you mean, you know, that we think some species are elevated above others. I do think some species should be considered more significantly than other species, so I'm out of kilter with that particular modern view, because otherwise I think everything disintegrates, I can say. You know, because if we considered an animal equal to a human being, you could become vegetarian. But since a plant is alive too, it's a species, we couldn't eat anything. So it becomes sort of untenable, I think. So I know not everybody agrees with that, but that's my view.
Paul, thank you. But I just have a question. Amongst
You're breaking your own rules.
I know, but it's a question on behalf of everybody. It strikes me that in this French-speaking audience, there's a more philosophical bent to the questioning you're receiving than we would receive if we were in, for instance, the UK. Do you think that's true? You give this lecture in different places.
I think philosophy is an important aspect of it. I think the French culture is more philosophical and metaphysical than the English culture. I do think that. And I think it's healthy to have different biases.
Thank you very much indeed.